Welcome back, friends. Welcome back again to one more other lecture session from this channel. So in this lecture session, we will be studying regarding one more other chapter from the NCERT, which is nothing but biotechnology principles and processes in detail. So it is one of the easiest chapter and even it is completely knowledge based chapter. And in this particular chapter, here we can expect nearly 3 to 4 questions in the NEET examination. So here prior to the explanation of the various concepts which come under biotechnology principles and processes in detail, at first let us have a look on the term known as biotechnology. So here what is biotechnology or how we can define the term known as biotechnology. So here the branch of biology which deals with the uses of organisms or microbes and even with the enzymes which were isolated from the organisms in order to regulate the production of many of the beneficial products to the human welfare. So here it is termed as biotechnology. And here basically biotechnology can be classified under two different classes, and these may include ancient biotechnology and the other one is modern biotechnology. Is it clear? So let me write it on the board. The branch of biology that deals with the uses of microbes or organisms and the enzymes isolated from the organisms in order to regulate the production of many of the beneficial products to the human welfare. So here it is termed as biotechnology. In order to regulate the production of many of the beneficial products to the human welfare. Production of many of the beneficial products to the human welfare. So here it is termed as biotechnology. And here, as already mentioned, here we can classify or here we can differentiate biotechnology under two different classes, and these may include ancient biotechnology and the other one is modern biotechnology. So let me write it here. Biotechnology, it can be classified under two different classes, and these may include ancient biotechnology and the other one is modern biotechnology. So at first, if we consider ancient biotechnology, so here what is ancient biotechnology? Basically, ancient biotechnology, it is a microbial mediated process. So here by taking the advantage of microbes, many of the beneficial products, they can be successfully synthesized here. So here some of the beneficial products which can be successfully synthesized by the means of ancient biotechnology, these may include production of curd from the milk. Is it clear? Some of the beneficial products like production of curd from the milk and production of cheese and even production of specific alcoholic beverages like wine and even preparation of bread. So here all these products, they can be successfully synthesized under ancient biotechnology. Is it clear? So let me write it here. Ancient biotechnology basically it is microbial mediated process. microbial mediated process which involves the production of many of the beneficial products like production of curd from milk, and even preparation of cheese. Is it clear? Preparation of cheese, bread, and even production of alcoholic beverages like wine. Production of alcoholic beverages like wine. So here all these products, they can be successfully synthesized under ancient biotechnology. And if you consider modern biotechnology, so here basically modern biotechnology, it takes the advantage of genetically modified organisms. So here by taking the advantage of genetically modified organisms, many of the beneficial products, they can be successfully synthesized at larger scale. So here some of the beneficial products which can be successfully synthesized by taking the advantage of modern biotechnology, it may include production of test tube baby by the means of in vitro fertilization. Is it clear? So let me read it here. Modern biotechnology. Modern biotechnology basically takes the advantage of genetically modified organisms in order to regulate the production of many of the beneficial products at larger scale. Many of the beneficial products at larger scale. Right? So here some of the products which can be successfully synthesized under modern biotechnology, it may include production of test tube baby by the means of in vitro fertilization. Is it clear? So let me write it here. Some of the beneficial products which can be produced under modern biotechnology may include production of test tube baby by the means of in vitro fertilization. And the other one is production of antibiotics. And the other one is enzymes and organic acids. organic acids. So here all these beneficial products, they can be successfully synthesized or they can be successfully produced at larger scale by the means of modern biotechnology. So here by taking the advantage of the above information into consideration, European Federation of Biotechnology, it has given a simple and standard definition for biotechnology. So here biotechnology, it encompasses both the traditional view and even the modern molecular biotechnology. And here it is an integration of both the natural science and even it takes the advantage of various components of living organisms like cells, tissues, organs and even the molecular analogs in order to regulate the production of many of the beneficial products to the human welfare. Is it clear? So let me arrange the things here. So here, as already mentioned regarding the standard definition which was given by European Federation of Biotechnology. So here biotechnology, it encompasses both the traditional views and even the modern molecular biotechnology, and it is an integration of both the natural science and even it takes the advantage of various other components of living organisms like cells, tissues, organs and even the molecular analogs in order to regulate the production of many of the beneficial products to the human welfare. So let me write it here. European Federation of Biotechnology had given simple and standard definition for biotechnology. So here biotechnology encompasses both the traditional view and modern molecular biotechnology and even it is an integration of natural science and various other components of living organism. Living organism like cells, tissues, organs and molecular analogs, in order to regulate the production of many of the beneficial products to the human welfare. Is it clear? In order to regulate the production of beneficial products to the human welfare. And if you look at the principles of biotechnology, so here what are the various principles of biotechnology? So here, out of many of the principles, the two core techniques that enable the birth of modern biotechnology, it may include genetic engineering and the other one is bioprocess engineering. So here, what is genetic engineering and what is bioprocess engineering? So let me write it on the board. Principles of biotechnology. Principles of biotechnology. So here the various principles of biotechnology, it may include genetic engineering. Genetic engineering and the other one is bioprocess engineering. Bioprocess engineering. So here what is genetic engineering and what is bioprocess engineering? So at first if we consider genetic engineering, so here genetic engineering basically involves in regulating the alteration or in regulating the change in the genetic material of a living organism and ultimately to change the phenotype of the host. And here it is termed as genetic engineering. Is it clear? So just let me write it here. Genetic engineering, it involves in regulating the change in the genetic material of a living organism and ultimately to change the phenotype of the host So here it is termed as what? Genetic engineering. So here the meaning of this particular concept we will be studying or we will be learning in the upcoming lecture sessions. And here if we consider bioprocess engineering. So here what is bioprocess engineering? So here bioprocess engineering basically involves in regulating or in maintaining the sterile environment, microbial free environment or contamination free environment in association with bioprocess engineering in order to regulate the growth of many of the microbes or in regulating the growth of many of the desirable eukaryotic host cells in order to regulate the production of many of the beneficial products to the human welfare at the larger scale. And here it is termed as bioprocess engineering. Is it clear? Bioprocess engineering, it involves in regulating the maintenance of sterile environment, microbial free environment and contamination free environment. For the growth of microbes and desirable eukaryotic host cells. In order to regulate the production of beneficial products, to human welfare at larger scale. So here in order to regulate the production of organic acids, enzymes, antibiotics. So here there is a requirement of sterile environment, microbial free environment or contamination free environment. So here it comes under bioprocess engineering. So here bioprocess engineering, it plays an important role in regulating the production of many of the organic acids, enzymes, antibiotics at larger scale. So here by taking the advantage of bioprocess engineering, production of all these products, they may take place at larger scale. Is it clear? So let me erase the things here. And if you look at the conceptual understanding of the principles of genetic engineering, so in general, we do have two different modes of reproduction. One may include sexual mode of reproduction and the other one may include asexual mode of reproduction. So here we can appreciate sexual mode of reproduction in comparison with asexual mode of reproduction. So here why we can appreciate sexual mode of reproduction in comparison with asexual mode of reproduction here? Because basically sexual mode of reproduction, it is responsible for regulating the variations and even it is also responsible for regulating the recombination in between the unique genetic setup among the living organisms or within the population. But whereas asexual mode of reproduction is most commonly responsible for regulating the preservance of the genetic material among the living organisms. Is it clear? So let me write it on the board. We can appreciate sexual mode of reproduction in comparison with asexual mode of reproduction. So here, how we can appreciate sexual mode of reproduction in comparison with asexual mode of reproduction? Here, sexual mode of reproduction, it is responsible for regulating variations and even it is also responsible for regulating the recombination in between the unique genetic setup among the living organisms or within the population. In between the unique genetic setup within the living organisms or within the population, right? So here, what is the meaning of this particular concept here? If the sexual mode of reproduction, if it has took place in between the two different partners who do not have any blood relation, and if the offspring has obtained from these two partners, in that particular offspring there is a higher chance for the occurrence of variation. Is it clear? And here, asexual mode of reproduction, most commonly it is responsible for regulating the preservance of the genetic material. Asexual mode of reproduction is responsible for regulating the preservance of preservance of genetic material among the living organisms. So here, traditional hybridization procedures, it allows the expression of both the desirable genes and as well as undesirable genes. So the reason why, in order to eradicate the problem, instead of taking the advantage of the normal sexual mode of reproduction, here we can take the advantage of one more other alternative strategy through which we can clone only the desirable genes but not the undesirable genes. And here the specific alternative strategy through which we can clone only the desirable genes but not the undesirable genes, here it is termed as gene cloning or gene transfer. Is it clear? So let me write it here. Traditional hybridization procedures. Traditional hybridization procedures allows the expression of both the desirable and undesirable genes right? So the reason why, instead of taking the advantage of the traditional hybridization procedures like sexual mode of reproduction, here we can take the advantage of one more other alternative strategy through which we can clone only the desirable genes but not the undesirable genes. And here it is termed as gene cloning or gene transfer. By taking the advantage of gene cloning or gene transfer, here we can clone only the desirable genes, but not the undesirable genes. So here, in order to understand this particular concept, here we can take the advantage of one of the common example. So here let us consider one specific host cell like E. coli. Is it clear? So here within the E. coli host cell, in addition to the circular chromosome, there are many other extra chromosomal double-stranded circular DNA molecules which are termed as plasmids. So here the particular plasmids, they act as vectors or they act as cloning vehicles. Is it clear? So let me show it on the board. So let us assume it as the E. coli host. E. coli host, right? So within the E. coli host, in addition to the circular chromosome, there are many other extra chromosomal double-stranded circular DNA molecules which are termed as plasmids. Right? So here these are nothing but what? Plasmids. So here these particular plasmids, they act as cloning vehicles here. So here, in order to clone the desirable gene, at first here we need to isolate the plasmid DNA from the E. coli host. Is it clear? In order to clone the desirable gene, at first here we need to isolate the particular plasmid DNA from the E. coli host. Plasmid DNA, right? So here, after isolating the plasmid DNA from the E. coli host, here we need to isolate the foreign desirable gene. So here, after isolating the foreign desirable gene from the particular source, here we need to cut the particular plasmid DNA at one particular desirable site by taking the advantage of a specific enzyme termed as restriction enzymes. Is it clear? Restriction enzymes, right? restriction enzymes. So here these particular restriction enzymes, they are site specific, and here these particular restriction enzymes, they are capable to cleave the particular plasmid DNA at one particular desirable site. So here let us be assume this is the particular desirable site, right? So this particular desirable site, it has cut by taking the advantage of restriction enzymes. So here after cutting the particular plasmid DNA by taking the advantage of restriction enzymes, here we need to insert the foreign desirable gene. Foreign desirable gene, right? So here it is nothing but the foreign desirable gene. Foreign desirable gene. Foreign desirable gene. So here once when we insert the foreign desirable gene within the particular plasmid DNA, now here the particular plasmid DNA, it has turned into recombinant plasmid DNA. Is it clear? After inserting the foreign desirable gene within the particular plasmid DNA, now here the particular plasmid DNA, here it is termed as recombinant plasmid DNA. Recombinant plasmid DNA. Plasmid DNA. So here most commonly the particular foreign desirable gene, it has to integrate with the particular plasmid DNA at one particular site which is very near to a specific site termed as origin of replication. Is it clear? The insertion of the foreign desirable gene within the particular plasmid DNA, it has to take place at one particular site within the circular DNA, and here it is termed as origin of replication. Origin of replication, right? Origin of replication, which is simply termed as ORIC. ORIC. So here let us assume it has the ORIC here. ORIC. So here, after the successful insertion of the foreign desirable gene within the plasmid DNA, now the particular plasmid DNA, it has turned into recombinant plasmid DNA. So here, after the successful formation of recombinant plasmid DNA, now here we need to insert, or here we need to place the recombinant plasmid DNA into the E. coli host. So here, after placing or after inserting the recombinant plasmid DNA into the E. coli host, now here, likewise the other genes in case of the plasmid DNA, even the foreign desirable gene, it also undergoes successful expression within the progeny cells by taking the advantage of a specific enzyme termed as host DNA polymerase. Is it clear? So let me write it here. Host DNA polymerase. Host DNA polymerase is responsible for regulating the expression of genes. Expression of genes. And here it is termed as what? Gene cloning. Gene cloning or gene transfer. Gene transfer, right? Is it clear? So then after we will be studying regarding the construction of the recombinant plasmid DNA. So here how the construction of recombinant plasmid DNA can take place, and who were the people who were responsible for identifying the recombinant plasmid DNA or for constructing the recombinant plasmid DNA for the very first time. Is it clear? So let me arrange the things here. And if you look at the construction of the recombinant DNA plasmid, so here the construction of the recombinant DNA plasmid for the very first time it was took place in the year 1972. So here Herbert Boyle and Stanley Cohen for the very first time they have designed a specific plasmid DNA termed as recombinant plasmid DNA by inserting a foreign desirable gene which is responsible for encoding antibiotic resistance. Is it clear? So let me write it here. construction of recombinant DNA plasmid. Recombinant DNA plasmid. Recombinant DNA plasmid. It took place in the year 1972. In the year 1972, by inserting foreign desirable gene which is responsible for encoding antibiotic resistance. Antibiotic resistance, right? So here, construction of the recombinant DNA plasmid for the very first time, took place by Herbert Boyle and Stanley Cohen in the year 1972. Is it clear? Recombinant plasmid DNA for the very first time was constructed by Herbert Boyle and Stanley Cohen in the year 1972. So here, the recombinant plasmid DNA which successfully produces within the E. coli host, it is independent of the circular chromosome. So the reason why all the genes which are a part of the plasmid DNA, they are capable to express within the E. coli host independent of the circular chromosome. And here, in order to regulate the expression of all the genes which are a part of the plasmid DNA in association with the foreign desirable gene, here there is a requirement of a specific enzyme termed as host DNA polymerase. So here host DNA polymerase, it plays an important role in regulating the expression of all the genes which are a part of the plasmid DNA in association with the foreign desirable gene. Is it clear? Recombinant plasmid DNA which successfully produced within the E. coli host is independent of circular chromosome. And here host DNA polymerase plays an important role in manipulating the copy number within the E. coli host. And here, in order to regulate the formation of the recombinant plasmid DNA, here there is a requirement of specific enzymes like restriction enzymes and as well as DNA ligase. So here, what is the role of restriction enzymes here? Restriction enzymes play an important role, play an important role in cutting the plasmid DNA, in cutting the plasmid DNA at desirable site. And here, after cutting the plasmid DNA by taking the advantage of restriction enzymes, here we need to insert the foreign desirable gene within the plasmid DNA. So here, after the insertion of the foreign desirable gene within the plasmid DNA, now here we need to ligate the foreign desirable gene with the plasmid DNA. So here, in order to regulate the ligation of the foreign desirable gene with the plasmid DNA, here there is a requirement of a specific enzyme termed as DNA ligase, which is responsible for regulating the ligation here. Is it clear? DNA ligase plays an important role in regulating the ligation in between the foreign desirable gene and the plasmid DNA. Right? So here, after the ligation of the foreign desirable gene in association with the plasmid DNA, here the expression of all the genes of the plasmid DNA they take place within the E. coli host. And here the specific mechanism or the specific process here it is termed as what? Gene cloning or gene transfer. Is it clear? Gene cloning or gene transfer. Now let us have a look on the various steps which are involved in regulating the formation of the recombinant plasmid DNA within the genetically modified organisms. Is it clear? So let me arrange the things here. And if you look at the various steps which are involved in regulating the formation of recombinant plasmid DNA within the genetically modified organisms, here there is a requirement of 3 different steps in regulating the formation of recombinant plasmid DNA within the genetically modified organisms. And these may include identification of, so let me write here, there are 3 basic steps which are involved in regulating the formation of recombinant plasmid DNA. Recombinant plasmid DNA within the genetically modified organisms. Within the genetically modified organisms. And these may include identification of foreign desirable gene from the concerned source. Is it clear? Identification of foreign desirable gene containing DNA from the source. And the next specific step here is nothing but what? Transfer of transfer of the desirable DNA from the source into the E. coli host. Is it clear? Transfer of foreign DNA from the source into the E. coli host. So here likewise the E. coli, the other host which is responsible for regulating the gene cloning or gene transfer, here it is nothing but what? Salmonella. Is it clear? Salmonella, it is also most commonly used as a specific host cell for regulating the gene cloning or gene transfer. And the last step here is nothing but what? Maintenance and production of desirable gene product within the progeny cells. Is it clear? So here all these are the various steps which are involved in regulating the formation of the recombinant plasmid DNA and as well as the expression of the foreign desirable gene within the progeny cells. Is it clear? So here this is regarding the introduction of biotechnology principles and as well as processes in detail. And in the next upcoming lecture session, we will be studying regarding tools of recombinant DNA technology in detail. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.